சிம்போனியின் இந்த பாடல்கள் உங்களுக்கு பிடிச்சிருக்கா? உடனே நம்ம சேனலுக்கு சப்ஸ்கிரைப் பண்ணுங்க. உங்கள் ஸ்கிரீனில் தெரியும் சிகப்பு பட்டனை அழுத்தி பக்கத்தில் உள்ள மணி பட்டனை அழுத்துங்கள்.
To start this auspicious session with prayer song. Dear God, give us wisdom to make this world a better place. To welcome us to Shohana, to welcome us to establish integrity. Welcome address is the most important speech in any function because it sets the mood of the function. To give a red carpet, welcome to everyone assembled here. May I take the pleasure of requesting my dynamic HOD sir, Dr.S.SUBBURAYALU, to welcome the gathering. Please, sir.
Thank you, Murgavini. Good morning to everyone present here and each one of you present today. With the blessing from the Almighty, I wish to express my sincere thanks to you all for giving me an wonderful opportunity to deliver my welcome address before you through this Google Meet and YouTube platform. On behalf of our beloved management, respected principal, heads and faculty members of various departments of our college and the faculty members of our own department, alumni, and the students of our department, it is a matter of great pleasure and honor for me to welcome today's chief guest and resource person, Dr. VASUDEVAN AYYAPPAN, research scientist, molecular genetics and epigenomic laboratory, College of Agriculture and Related Science, Delaware State University, Delaware, United States of America, who has given his consent, even his busy schedule, to talk on the science of epigenetics in the occasions of international webinar. It will be conducted as a part of 25th year celebration of our college, conducted by Department of Biochemistry. Moreover, you can ask me, dear participants, you can ask me, you have a doubt on why we have selected this topic for today's discussion. This topic is very, very interesting. You know that epigenetic changes begin before you are all born. All of your cells have the same genes, but act differently, isn't it? As you grow and develop, epigenetics helps to determine which function a cell will have. For example, whether it will become a heart cell, a nerve cell, or a skin cell. Such a interesting and informative discussion today we have with Dr. VASUDEVAN AYYAPPAN. Moreover, Dr. VASUDEVAN AYYAPPan, a research scientist, son of our soil, belongs to Putti, Japan in Tamil Nadu. He is an eminent and enthusiastic scientist, and he is highly studious in his all academic contributions, such as served as professor, postdoctoral researcher, research scientist, and a scriptures of innovations idea into a scientific thoughts like that. I know him for past 2 years only, but when I approached him to give a talk for this international webinar, immediately he accepted my invitation, even his busy schedule. He has earned a very good name among the staff and students of educational and research institutions wherever he served. He never tired till the students to understood the delivery content of him, both in theoretical classes and technical classes, means imparting laboratory techniques such as RT-PCR, ELISA, STS batch, HPLC, Western blotting, etc. into the minds of young scientists. Such a great, active, dynamic personality is our resource person, research scientist, Dr. VASUDEVAN AYYAPPAN. On behalf of our management, principal, and entire administration of our college, we welcome you, sir. I welcome Dr. VASUDEVAN AYYAPPAN. Welcome you, sir.
Thank you so much, sir. Thank you so much for the welcoming.
Thank you, sir. Thank you, sir.
Yeah.
It is also my immense pleasure to welcome our most respected and honorable principal of our college, Dr.S.MATHIVANNAN. He is an active and a dynamic personality. He is a great academician, good administrator, and knows nook and corner in the administration. He has served as professor and head of the department in the discipline of commerce for more than 35 years in S. Ramasamy Naidu Memorial College, Sattur, a reputed and a pioneer institutions in South Tamil Nadu. He is a man of actions, conducted many more symposiums, seminar, conference by getting funds from various funding agencies in India. And recently he has conducted a mega alumni meet in the name of ComConnect 2019 when he was in SRNMC. Academically, he is a brilliant and produced more than 10 PhDs under his guidance. Most of the students of him working in various reputed universities and institutions in India as well as abroad. Even though he was well settled, he always till now dedicated his life for the betterment of the students coming from rural areas in and around Kovilpatti, and always motivating them to uplift their lives. Such a great personality is our principal, Dr.S. Mathivannan. On behalf of our entire participants, I welcome you, sir. Sir, welcome
Thank
you,
you,
sir.
Subbrayalu. Thank you, Subbrayalu.
Thank you so much, sir. I also extend my warm welcome to heads of various department and faculty members of our college who motivated and encouraged us to conduct such a type of interactive webinars and symposium, and also provided support to conduct this webinar in successful path. On behalf of these, because of these participants, I also welcome our faculty members. It is also my duty to express my heartiest welcome to faculty members of our own department, Dr. Bhuvaneswari Sandara, Mrs. R. Sivasangari, and Dr. L. Vishnuvardhini, and Mrs. Vanilla, who have provided utmost support to organize this webinar in a grand manner. Last but not least, I welcome alumni of our students of participating institution and students from our department who are going to learn some important scientific thoughts in the science of epigenetics and in this auspicious occasion. Once again, I welcome you all for this international webinar on the science of epigenetics. Welcome you all. Thank you for giving an opportunity to welcome resource person and guests of our this international webinar. Thank you.
Thank you, sir, for your pleasant welcome, which boosted our mind. All of us do not have equal talent, but all of us have an equal opportunity to develop our talent. Famous saying by former President Dr. APJ Abdul Kalam. Now I kindly request our principal, sir, who gave this great opportunity to improve our talents, to deliver the felicitations. Please, sir.
Thank you, ma'am. Thank you. The most respected resource person of this day, Dr. Vasudevan Ayyappan, faculty members, and HODs from participating institutions, HODs, faculty members, and students of our own college, alumni of the Department of Biochemistry of our college. Good morning, you all. It is my privilege to felicitate and to wish the participants of this international webinar on the science of epigenetics, which is organized by the Department of Biochemistry as part of the 25th year celebration of our college. This wonderful program is arranged particularly to the young scientists of our college and from various colleges to discuss about the concepts, fundamentals, and the recent developments behind the science of epigenetics by our distinguished resource person, Dr. Vasudevan Ayyappan, who is serving as research scientist in molecular genetics and epigenetic laboratory, College of Agriculture and Related Sciences, Delaware State University, United States of America. The scientist Dr. Vasudevan Ayyappan, having more than 20 years of teaching experience and published a number of research articles in peer-reviewed journal and in reputed scientific journals at international level. And his field of interest are plant virology and functional genomics, molecular epigenetics, molecular oncology, cellular and developmental biology, DNA sequencing, and genotyping, and etc. I came to know that the great scientist, that is our resource person, has contributed a lot in the field of molecular genetics and epigenomics through Dr. S.SUBBURAYALU, head of the Department of Biochemistry, during our discussions about the resource person in many occasions. I am sure that the experience and the expertise knowledge of our resource person will be much useful to the participants to understand the concepts of today's discussion on the scientific epigenetics. In biology, the epigenetics is the study of heritable phenotype changes that do not involve any alterations in the DNA sequence. It has many and varied potential medical applications. In 2018, the National Institute of Health in India announced that $190 million has been earmarked for epigenetic research over the next 5 years. It indicates that the epigenetics has the potential to explain mechanisms of aging, human development, and origins of cancer, heart disease, mental illness, as well as several other conditions. I take this opportunity to welcome and thank our resource person, Dr. Vasudevan Ayyappan, who has accepted our invitation during his busy schedule to share his knowledge and research experience in the field of molecular epigenetics. On behalf of our management, faculty members of our college, I welcome the resource person and all the active participants, and also I wish a grand success of this webinar. During this scientific session, our resource person will highlight the fundamentals and innovations behind the field of molecular genetics and epigenomics among the budding scientists, that is students and research scholars. I hope this webinar will be highly informative and interactive to share the ideas and the secrets of the scientific thoughts of epigenetics. The great scientist Albert Einstein says that a scientific forum would exhibit the secret of God's thought. I am sure that this international webinar will also reflect the thought of Albert Einstein. Finally, it is my duty to appreciate and congratulate the head of the department and the faculty members of Biochemistry department of our college for organizing such a wonderful international webinar for the benefit of our students and faculty members from different institutions across Tamil Nadu. I take this opportunity to wish the alumni of the biochemistry of our college. Let me conclude by wishing that each one of you will carry back from here some vital scientific thoughts and will flourish you in the galaxy world of research. Good luck for you all. Wish you all the best. Once again, thank you, Nanda.
Thank you, sir. You have given very good information and informative talk you have given, sir, rather than a biologist, sir, regarding epigenetics. Thank you, sir. Thank you so much.
Thank you.
Thank you, sir, for your useful information about the epigenetics. At this point, may I call my friend R.B. Muthu Mahodharini, third year of BSc Biochemistry, to mention Dr. Vasudevan Ayyappan sir achievement and contribution and state reason for having her as a chief guest.
Be yourself and look for the magic in every moment. A cosy good morning to all those brisk and crisp observers gathered for this international webinar on the science of epigenetics organized by the Department of Biochemistry. It gives me immense pleasure to start this auspicious occasion by heartily welcoming our principal sir, all faculty members, and my dear fellow mates. Now, it's my great privilege and honor to warmly welcome our resource person, Dr. Vasudevan Ayyappan sir, a prominent scientist currently working at the Molecular Genetics and Epigenetic Laboratory at USA. I cherish and thank wholeheartedly each and everyone who has put equal efforts behind organizing this webinar. With this, I would like to honor our resource person and invite him delightfully to share his visionary thoughts over the topic and along with the guidance to the students. Our resource person, Dr. Vasudevan Ayyappan sir, has completed his UG in Botany, PG in Microbiology at MK University, and sir has also completed his PhD in Plant Biotechnology. Sir has done about 25 research publications, out of which one has appeared in Journal Highlights and another has appeared in the list of top 25 articles in Science Direct. Sir's teaching and mentoring experience are admirable. He had been an adjunct faculty at Trovan University, teaching microbiology for the senior level undergraduate students, as a co-instructor in the College of Agriculture and Related Sciences at Delaware State University to teach molecular genetics and genomics. He had acted as a full-time instructor in Department of Biology during the year 2006 to 2007. He is a teaching constant in Department of Biotechnology at Bharathidasan University. Dr. Vasudevan sir's teaching has widely extended in so many fields like genetic engineering, microbiology, plant molecular biology, genetics, genomics, general plant biology, and also in plant vaccines. Scientific skills and techniques are well versed in molecular biology, biochemistry, cell biology, sequencing and analysis, bioinformatics, and laboratory management. Sir has been a part in 27 conference proceedings and presentations, which are familiar with his stature, contribution, and recognition. His whole place as a demonstrator and organizer in so many biological workshops. He has also served as a member in Desertion Advising Committee and as an editorial member in Indian Journal of Natural Sciences, Journal of Drug Delivery, BBE, and so on. A research scholar, is eminent and widely known for his achievements. We are very grateful to you, sir, for accepting our invitation and making your presence here. Once again, I welcome you all. Now it's my moment of bliss and delight to cordially invite our resource person, Dr. VASUDEVAN AYYAPPAN, sir, for today's international webinar.
Yeah, thank you, Muthu. Thank you for everyone. I'm very glad to give the talk today. It's for just the general discussion. It's my good opportunity to talk to you, everyone. That I remember my olden days. That when I went to college in Savagasi, is kind of a name. Academic college called Anjak. That I did both my bachelor and master. So my native village is nearby the college. 4 kilometers from there, and this is a golden time. So it's like same thing, same like you, what you sitting there in the call. So you visit a lot of seminars, that days, during bachelor and masters, working with the different faculties at the time, so anybody can achieve these kind of things, like me, you stay in, you can work in different countries and you can serve to your own country, our country, India, all those things. So it's a good opportunity to, after my master, I did PhD in Tiruchirappalli in plant biotechnology, working on the aggregatory mediated transformation. That time, that how to grow the plants, genetically modified plants, and getting the good traits of that, the characters. So I'm really glad that as all our students, so you are like my students. I can ask any questions even after the presentation. You can send the email at any time. Okay. So with that, let's start the presentation. So can you able to see, sir, my presentation?
Yes, sir. We are able to.
Yes, sir.
Okay. Okay. So today's topic is the science of epigenetics. Okay. So here I given my email address. Okay. So you can send the email, or you can ask Dr. Subburayallu, sir, for my email address, and he can provide that one. Okay. So what's epigenetics? Epigenetics is a good topic. It's a vast, a broad topic. So epigenetics is including the human epigenetics, animal epigenetics, plant epigenetics. All those things is combined. Depending on which field we are working on. Okay. Currently, I'm working on plant epigenetics. My research is that how the plants are, the stress impact in the plants. Like stress means like adiabatic stress and biotic stress. In adiabatic stress, heat, drought, salinity, all those things. And the biotic stress, the fungi, how it impacts the plants and how the plants survive. There's a stress mechanisms. Okay. So now you know that epigenetics is a group of heritable modifications without alterations to the DNA sequence itself. Okay. So it's a natural mechanisms. There's no mutation here, but based on the environmental stress, our body changes automatically. That is the epigenetics. Okay. Epigenetics derives from the Greek word called epi meaning above. Okay, above. And when linked to the genetics, epigenetics is on top of genetics. It's including, the adding layer of the heritable characteristics in addition to genetic inheritance. So in the normal genetics, the classical genetics is like a genetic inheritance. Okay, here the heritable characteristics. It's called transgenerational transformation. The transgenerational, the characters to the coming generations. So epigenetic changes occur in like 3 ways. Okay, the 3 mechanisms, that is the modification to DNA bases, the methylation of cytosines, for example. It could be like acetylation also, cytosine also there. Okay, and alterations to histone proteins, that is by methylation or acetylation. Okay, and in general, remodeling of chromatin modifications, remodeling against the structure of chromatin. Okay, so there is this also involved the miRNA, microRNA changes, so long non-coding RNAs. So these 3 things are combined with epigenetic changes. Okay, it could be either of these. Okay, is leading to the changes in gene expression profile. Okay, and here, the cells have the same genes when you are younger, but the expression of genes are different when we grow. Okay, so epigenetics is the main thing is determine which function a cell will have. For example, whether it will become a heart cell, nerve cell, or skin cell. So when we grown up, this epigenetic modification changes. This leads to the cell. What is the function of those cells? They lead to the tissue and the gene expression changes. So that epigenetic modification is happening in every tissues. Okay, and here the epigenetics. If we ask questions, is the simplest difference. Okay, if genetics is genetics is the classical one. Is a mutation in DNA sequence. The mutation, like the DNA mutation, like methylation kind of things. Epigenetics is that including DNA methylation, long non-coding RNAs, ncRNA interactions. It could be miRNA or siRNA, and histone modifications, methylation and acetylation. So these, the end of that, the changes, the change in the phenotype. The phenotype, this could be like for example, the obese or the have the problem in the growth, all those things, malnutrition, everything. So epigenetics and the genetics both lead to phenotype. Genetics, mutation in DNA sequences, there is the mechanism happening by chemicals modifications. But in epigenetics, it's natural mechanisms happening by these 3 pathways. When you see here, the environment is the one leads to epigenetic modifications. There are gene regulations, okay, lead to phenotype. The same thing, genetics, okay, the DNA sequence, the variations, okay, there is the mutation lead to gene regulation. So in both cases, the phenotypic changes. Okay, so our body is changing slowly by the environment. That is the epigenetic modifications. When you see here, exposome, epigenetic process, and the phenotype. Right? The exposomes is including parental stress, maybe in the fetal, when the mom became pregnant, all those things, that the baby growing in the uterus. Okay, maternal behavior, how the mother behaving, the smoking or drinking when she become the pregnant, all those things, and nutrition, what kind of foods we are taking, chemicals and pollutants. So when you're traveling, when you're driving, what kind of chemicals pollutants going to your body, and the genetic variation. Okay, microbiota, disease pathogens. Okay, what is the microbes? Are you eating that good microbes or bad microbes? Okay, environmental stress. Okay, the heat, cold, climate, all those things, and management the practices, all those things. So it leads to the epigenetic processes. It could be DNA methylation, histone modifications, chromatin remodeling, that is chromatin positioning, and non-coding RNA regulations, including miRNA, siRNA. Okay, and finally to the phenotype. The phenotype, for example, the structure of meat changes, and reproduction cells changes, and the eggs formation. Okay, so all those things, even the cow also, the milk we're getting the cow, the modification in the what kind of food, the feed, the cow is eating. Okay, and disease resistance, the plants resistant to the disease. It's also epigenetic modifications. Okay, there is natural mechanisms and growth development, wool, all those things. The examples are the phenotype. Okay, it could be in the humans or animal or the plants. All those things are examples are here. So epigenetic changes are all cells contain the same genes, but the gene expression patterns are different in different cells. How? Because epigenetic modifications. Okay, every cell has same genes, but which genes are on or off in different tissues is very important. Different cells are important. Okay. So now epigenetic mechanisms contributing to gene regulation. Okay, it's including the DNA methylation, histone modifications, and non-coding RNAs. So all leads to the gene expression. Okay, we'll see one by one. Okay, now we see that the entire structure of the chromosome in nucleus, the cell nucleus. The chromosomes is a complex structure. Okay, it has histone DNA packing. Okay, the thing is that these histones are proteins surrounded by the DNA, the helix. Okay, so now this histone has the tails. So in those tails, the epigenetic factors are binding. Okay, that leads to epigenomics changes. Okay, and here histone tails may be the methylation, the methyl group added, the ME, ME3, or acetylation, AC. ME or AC will bind, will lead to it determine the function of the DNA, determine the expression of the gene expression. Okay. See now what happened here? The epigenetic mechanisms, okay, it leads to the various environmental factors. For example, development, including uterus, childhood development. Okay, how the child grow, which environment grows, very important. Environmental chemicals, the pollutants, all those things, drugs, pharmaceuticals, activities, what we are taking the drugs, aging, diet, all those things will affect, the DNA structure, the chromosome. Okay, see now this helical structure, it has the DNA, and the DNA is surrounding the histones here. Okay, the histone tail has that the binding factor is called the epigenetic factor. Okay, so this DNA will be active or expressive. Okay, when it is like that, the free mode, gene active is like when the DNA accessible, that means the acetylation take place. Acetylation. Okay, but in some case, the middle diagram, the box, that here the proteins are tightly compact to the DNA. Okay, now in this case, DNA cannot be expressive. DNA, the gene inactive. Okay, DNA inaccessible and gene inactive. That means like there is no like the transcription and translation. Okay, so depending on the structure of the histones, depending on the activity of histones, how the histones are allowing the DNA to express. Okay, there's no. So in the tightly compact, that means like methylation, herpes, histone tail, and in the case of here, there is acetylation taking place. Okay, that means like the DNA can able to express. That means the lead to transcription. Okay. So now is epigenetic modifications alter which genes are on or off. Okay, it's like freely arranged DNA, and here the tightly compact of the DNA. Okay. See now the DNA modifications, chromatin modifications, and non-coding RNAs. Okay, and like finally the RNA modifications. Okay, and here the normal RNA. Okay, so here the cytosine methylation and hydroxy methylation, where it could be methylation in the cytosine area is DNA methylation, the first case. Okay, and adenine methylation, both case. Okay, C and A methylation, the first one. Second thing is that chromatin modifications. That means like histone variants, the variants of histones. The histones have that 8 subunits: H1, H2, H3, and H4. Okay, in each subunit has, each histone has 2 subunits. The total 8 subunits of histones. Okay, and here the histone modifications, the positioning of the histones is important here. Okay. Third thing is that long non-coding RNAs, it is like a miRNAs. It's a non-coding RNA. So you know that RNAs will be coding regions, right? But here long non-coding RNA, there's no coding regions. That means we can't get the complete protein structure after the translation. Okay. And here mRNA and RISC, there's an RNA-induced silencing complex. It breaks the RNA into miRNA. That is like a non-coding RNAs. And finally, RNA modifications. The RNA modifications, the poly A tail, there is RNA not able to the translate into protein. There's the inhibition of RNA into the protein. That's the final thing. Okay, so these 4 things are common epigenetic modifications in every single cells. Okay, so now these the simple example is DNA methylation. The top one. So M means methylation. It could cytosine methylation or adenosine methylation. And here the DNA is here, and you have the histones, and this one is the histone modifications, and it's methylation and acetylation. The purple color is that methylation, and green color is the acetylation. Okay. When you see here, there are 3 things. One is that DNA methylation, the bottom one, and the histone tail has that methylation has place and acetylation has place. Methylation and acetylation in the histone tail. Okay, this is the DNA. Okay, so DNA methylation and histone modifications, including methylation and acetylation. Okay, so in the case of child development, okay, the child has the epigenetic modifications is taking place when the child in the mother's uterus. Okay, there is that environmental influences. Okay, and children's experiences when they grow actually affect the expression of their genes. So during that, the development of the fetus and the early development of the child. So both are important, for getting the child when the child grows at the age. So very important that we have to take care of the child very carefully. So when they grow, okay, even then when they they're formed developed in the uterus. This means the whole idea that genes are set in stone. That means the genes are those are genes, and they express only in the particular place, is not able to express more. Those concept is no more active. Okay, so now the nature or just nurture, any one of these is no longer is a defect. Is nearly always both. Nature important, also nurture important. That means the nature should be good, and also the nutrition. When the child grow, we have to give the good nutrition in order to get the genes properly expressed. Okay. During development, the DNA that makes up our genes accumulates chemical marks. Here, chemical marks including the DNA methylation, the histone modifications, all those things that determine how much or little of the genes is expressed. So those histone mark, chemical marks or histone marks, it will determine whether genes are expressed or suppressed. So this collection of chemical marks is known as epigenome. The collection of chemical marks is called epigenome, and throughout the whole genome epigenome. Okay, so that we are doing the sequencing is called a next generation sequencing. That's called it will give the whole epigenome. That's what my research involved. Okay, and I published a lot of articles on it. I'll show you at the end of the presentation. Okay, the different experiences children have rearranged those chemical marks. So it will erase the chemical marks, depending on the which environment the child is growing. Okay, so because of the different experiences the children have, okay, it leads to the epigenetic modifications, so that the twins can exhibit different behaviors, skills, health, and achievement. Even though they grow in same environment, but the slight modification there, how they are behaving. Okay. And this epigenetic modifications, epigenome, they're happening in different ways. There are environmental factors, including the smoking, obesity, age, diabetes, all those things. Okay, they lead to the, hypertrophic cardiomyopathy, that the heart, all involving heart disease, heart failure, atherosclerosis, vascular dysfunction, and impaired angiogenesis, all those things. Okay, it's due to the not only the aging, it's also involving smoking, drinking. So we think that our body is good, we are doing good, even though we are taking some, the drinks, all those things, smoking, but our body is slowly changing and modifying so that one day we'll get the heart disease. So we are getting the heart problem. Okay, because the thing is that the slow changes in the cell level, the gene expression changes, and finally one day, something happens. Okay, so now the generation. So the generational, the inheritance, the transgenerational inheritance. So a woman who smokes during pregnant induces epigenetic changes in 3 generations at once. In herself, in her unborn daughter, and her daughter's reproductive cells. It's not only the mom, the mother, is for her children and children's children. Okay, so that it leads to different generations. So you're very careful, when we are doing kind of things. Smoking is not only affect the mother, it also affect the other 2 generations. Okay. And environmental factors, the first generation mother and father, it leads to the second generation fetus. There is her child, and the third generation, the fetus germline. That is so the thing is, when the fetus grow at that time, there is a changes in the epigenetic modifications, epigenetic changes. So those changes will lead to the future generations. That is her grandchild. The transgenerational inheritance. That is the role of epigenetics. It is applicable for both the human, animal, and plants. Okay, so my research involving in the transgenerational inheritance in plants. So we give stress to the plant, okay, and we collect a seed from that plant, and we grow the plant, in different, like a season, and we see how the the gene expressed, for the same stress. And again, we collect the seeds from second generation plants and grow the seed, okay, and get the plants and see that what is the epigenetic modifications that are taking place, all those things. So same thing applicable for the us as a human being, okay. See now, this that environmental factors like air pollutants, radiation, endocrine disruptors, hypoxia, diet, tobacco, alcohol, metals, and also there is the recent development in research called nanoparticles. These nanoparticles, that are also involving in the, that changes the epigenome. So it's like, it is not well studied yet. Okay. Okay, now they see that the examples of epigenetic modifications, the alterations in the cancer cells. Okay, so in cancer cells, we compare normal cell, how is like taking place? What is the reason for the cancer? What exactly happening? Okay, here, for example, the first case is DNA methylation. In DNA methylation, the gene promoter for the normal cell is same like, the TSS, that means transcriptional start site. That means where the transcription starts, or TSS. Okay, so this gene promoter is active as a normal, so that the DNA to give RNA to protein. The transcription and translation takes place. But in the cancer cell, what happened? The gene promoter, where there is a hypermethylation. methylation taking place in the gene promoter of tumor suppressor genes. So the tumor suppressor genes is the one to suppress the tumor. Okay. So now what happened if the promoter of the the cancer cell, there is the tumor suppressor cells, what happened? This promoter cannot, the DNA cannot be active to suppress the cancer cells. So it leads to the the tumor development. Okay, so the tumor suppressor genes are no more active because of the hypermethylation of the promoter region. Second thing is that histone modifications. That means including the methylation and acetylation on the histone tails. Okay, there is a loss of H4K12, K20 Me3 and H4K16 AC acetylation. Here, H4 means histones H4. K means amino acid position. K20, K means that there is the lysine. Okay, the lysine in the 20th amino acid positions. Okay, and Me3 means the 3 molecules of methylation. Okay. So now what happened that the loss of H4K20 Me3 and H4K16 AC leads to the tumor development. Okay. The third thing is that non-coding RNAs like miRNA and long non-coding RNAs, okay, including siRNAs too. And here there's up and down regulation of miRNAs, okay, in the target oncogenes and the tumor suppressor genes. So now what happened that the upregulation of miRNA, okay, in the oncogenes leads to the cancer, and downregulation of that miRNAs, they target tumor suppressor genes, that leads to the cancer. Okay, in both cases, the cancer development. The upregulation of miRNAs, the target oncogenes, okay, oncogenic miRNAs, upregulated, and downregulated tumor suppressor genes. So in this case, there is the miRNAs will be downregulated. Both leads to the cancer cells. Okay, so these are the 3 fundamentals. The reason that how the normal cells leads to cancer cells. Okay, so any questions, please let me know at the end of the presentation. It's very important to understand how the normal cells will become the cancer cell. How the person getting the cancer. Okay, there is a natural changes in the body. How these the cells are hyperactive. The genes expression. It could be the loss of methylation or acetylation, or or the hypermethylation. Okay, or the down or upregulated of miRNAs. Okay, so now the oncogenic expression. Here, the global changes in the epigenetic landscape are a hallmark of cancer. The global changes in the epigenetic landscape. It could be 3 mechanisms. Okay, either the DNA methylation, histone modifications, or miRNA, long non-coding RNAs. And disruption of epigenetic processes can lead to altered gene function and malignant cellular transformation. The disruption of epigenetic processes. Okay, and abnormal epigenetic modifications can contribute to tumorigenesis and resistance to cancer therapy by activating oncogenes. So here, in this case, the oncogenes are, onco means cancer genes. The oncogenes are activated. How? Abnormal epigenetic modifications. Okay, so we have the normal and abnormal epigenetic modifications taking place in our body. Okay, if everything okay, everything is going good, normal epigenetic modifications, that's fine, because the normal means when you're growing from childhood to that the kid and boy, adult, called kind of things getting aging, right? Aging is normal epigenetic modifications. But abnormal means like getting cancers, all those things. Okay, activating oncogenes and deactivating tumor suppressor genes and genes involved in the immune response. Okay, the tumor suppressor genes are the one will suppress the tumor. But now deactivating these tumor suppressor genes, what happens means these genes are no longer active. Okay, and the genes are no longer active. That means to the cancers. Suppressing terminal differentiation. Okay, and affecting tumor heterogeneity. So all those factors are contributing how the normal cells become the tumor cells. Okay. So now what is the solution for this? Okay. How we can eradicate this cancer cells? By epigenetic modifications, epigenetic way. Okay. Therapeutic potential. Okay. The pharmacological research in this area focuses on approaches to cancer treatment that include inhibiting the epigenetic modifications. Inhibiting the abnormal epigenetic modifications. Okay. What are the epigenetic modifications involved in histone modifications, chromatin remodeling, and transcription regulation? Okay. Strategies for addressing epigenetic modifications therapeutically include targeting writers, erasers, and readers during the transcriptions. Okay, as well as other pathways that regulate gene expression, that is such as PERP and BET. So these two pathways are very important in the cancer development. Okay, when I worked in the South Korea in the cancer development, in cancer research, I worked on this PERP and BET, the pathways. Okay, there are so many genes are involved in these pathways. Okay, so this epigenetic modifications, guys, is a complex mechanisms. It's not like simple mechanism. It's complex. It's a drastically the faster growing field. Okay, so especially in the cancer research. Okay, so how can eradicate this? The cancer cells in therapeutic ways. The first one is PERP inhibition, the pathway. So this pathway leads to cancer. Now inhibit the pathway. What happened? May lead to the accumulation of single and double-stranded DNA breaks in tumor cells. Okay, the tumor cells also has double-stranded DNA. So now we can break the DNAs that may have limited capacity for repair. Okay, the cells cannot able to repair or are exposed to DNA damaging agents resulting in apoptosis. Apoptosis means cell death. You know that. Okay, and BET family. There's BRD2, BRD3, and BRD4 and BRDT. This all the four combined, that's the family. Okay, the BET family protein inhibition can impair tumor cell, DNA transcription and induce apoptosis. So in this case, when you inhibit the BET family, that leads to the, it will suppress the DNA transcription. Okay, so that ultimately lead to the apoptosis. Okay. These are the important findings, the research still going on, in how we can eradicate to inhibit the cancer by inhibiting the pathway PERP and BET. Okay, so now let's say obesity is the main thing. How the person getting obese. Obese is a things is not only for the person eat more food. So more than that, a lot of things involved here. Okay. Genetic variation. So the environmental factors including diet, physical activity, chemicals, stress, all those things lead to obese. Okay, the person should need to the exercise. Okay, and eat the proper food. Take less calorie food and less sugars, all those things. Okay, and also expose to the chemicals. So now epigenetic modifications take place, like the DNA methylation, modification of keystone tails, and non-coding RNAs. Okay, we saw the previous slides that there is the detailed one. There is the DNA methylation, keystone modifications, okay, and non-coding RNAs, all the involved, right? The hierarchy structure is called mRNA. That means mRNA means microRNA. That there is the break. There is the hierarchy structure in the the long non-coding RNAs. So these two involving in the the RNA cannot be able to produce a proper protein. Okay, changes in gene expression. Okay, so what happened is it induced the obesity related genes. Okay, so when you're free, to read the articles, okay, what are the that obesity related genes, okay, that contribute to obesity? What is the epigenetic mechanisms involved there? Okay, so due to the time constraints, we don't have time to discuss more about it, and we will discuss in later. And here that our food also very important, guys. What kind of foods we're eating. It's like so more the healthy food or the bad food, like more fatty food like that. Okay, here the harmful food, in the westernized diet, where you live now, is burgers. So mostly that the fast food. The people want to eat fast. They don't want to cook like what you do in Indian culture. Okay, in the right side you see here there is the safe food called Mediterranean food, the diet. Okay, and here what is that? The obese phenotype and here lean phenotype. Obese phenotype, the fatty liver disease. Okay, accelerated aging. There is the age getting more faster. Okay, and hormone differences. Yeah, the techniques is basically the mainly the diets what are you eating. Okay. The main diet.
Already told, the diet technology means starvation tools is a very important one, isn't it, sir?
Yes, starvation. Yeah, the tool like the thing is that how much you eat is not important, and how we are eating. Like what is the frequency of eating, and also that if you eat like a little bit like 6 times a day is different, different than eating 3 times, eating 2 times more that what are eating. And also, what kind of food is fast food or like like natural food, all those things. I'm glad that now our food system is really good compared to Western culture. What we're going through right now. So the diets also very important, sir. How much you're eating and what kind of food you're eating, and also exercise.
Basically, we know that South Indian foods are having richest source of antioxidant, isn't it, sir? Antioxidant is one of the common. It's one of the factor is responsible for delaying aging.
Yeah.
Okay, sir.
That's true.
Sir,
and also now you're drinking more water. All those things. That thing is like
Okay, sir.
Yeah.
Sir, can epigenetics be applied in animals, sir?
Yes, sir. Epigenetics have animals going on, right? The Drosophila, they're working on studying that, and the yeast. Nowadays using mice also using that one. So like the thing is that the animals is the mice is the what do you say that? For they use the example, that the study using the mice, right? Whatever studies before going to the human being, they use the mice as the that model animals.
Model animals, yes. Yes.
Model animal for that. Definitely, that even the cow also, how the milk production, all those things, and the meat, the meat production, like all those things. Now they're working on that animals.
What about microorganisms, sir? Epigenetics is also applied to microorganisms, sir.
Yeah, definitely, sir. Microorganisms, for example, in the plants, right? The fungus. There is the natural fungus that is for the used for the there is benefits to the plants, right? Beneficial fungus, mycorrhizae. They close attached to the soil, right, and the plant root. So now, while doing that, they are sequencing the fungus. Mycorrhizae, and how they interact with the DNA with the plants. What's mechanism for that? They're doing this is going on right now. Okay, and several microbes are involved in including bacteria and yeast. They're doing they're using that epigenetics going on, right? So if we are working in the plant, okay, there are a lot of going on the microbes. So epigenetics is a growing field, and there's more opportunity.
Yeah.
Yeah.
Thank you, sir. Thanks so much, sir. Another one question from our Muthu Mahadarani, final students of our department. Are epigenetic modification heritable? Heritable, sir. It is inherited.
Exactly. Heritable, sir. Heritable. We see that in the previous slide, that how that mother smoking, it can lead to the the fetus, and also the fetus, the children's children also it will impact. Okay. So this is proven. It's incredible, sir. It's a heritance only.
Oh, thank you, sir.
Yeah.
Thank you. Thank you, sir. We are taking a lot of your time.
Welcome, sir.
Thank you, sir. Okay, sir. Oh, is there
Welcome.
any other questions? We will send to your mail ID, sir. Please clarify our doubts, sir. Okay, sir. Okay.
Definitely, sir.
Really,
So
we are happy, sir, to have a nice interaction with you, as well as we are learning very lot of information regarding epigenetics and its application in the field of medical also. Okay, sir. Thank
Yes,
you
sir.
so
Thank you,
much,
sir.
sir. Thank you so much, sir.
And any articles you need, just email. My email address is there in the presentation. Okay.
Nice, sir. Nice,
I can share this. And like any questions, students ask me other questions, then send me the information.
Okay,
And I'm glad to help you.
okay.
And if anyone is like want to in future studying in abroad,
Yes, that's why I'm asking you, sir. That's why I'm going to ask you, sir. Okay.
okay,
We should help us, sir, because we are in rural areas and students are coming from in and around Kovilpatti. So you must help them, sir. You must help them, sir. Thank you.
definitely, how to come to the master here,
Yes, sir. Yes, sir.
study master, and we can help. I can connect the person, who can sponsor visa, take care of all the documents,
Yeah.
we can talk one-on-one. Like anybody interested in, just send me the offline message, the email. I will talk about it. And America is that good for doing research, scientific research.
Yes, sir. Yes, sir.
Yeah, I'm glad, sir. I'm glad that the opportunity, and I feel like connecting our students from rural area, right? So that a student from the college,
Yeah.
the college, I'm glad that
Yes,
to
sir.
discuss everyone. Thank you, sir. Thank you for you and your principal, sir.
Principal, sir. Okay, sir. Sir, principal, sir. Principal, sir.
Maybe I think busy.
I think sir is having a lot of work on table. That's
Yeah. Definitely,
just a minute, sir. I think sir is attending this program. I think. Okay. Now I ask him, sir. Principal, you can, right? They are our principal.
Sir, otherwise, okay, sir.
Oh, yes, sir. Sir, if you are having, when you come to India, you must come to our college, sir. If we want to
Definitely,
honor you, our principal already told me for 1 week before, he told me, when Vasudevan come to Sivagasi, that time we should ask him to come out to our college. We have to honor. It's our duty. Sir already told me, sir.
No problem, sir.
சிவகாசிக்கு வரும்போது கண்டிப்பா எங்க காலேஜுக்கு வந்து எங்க ஸ்டூடெண்ட்ஸோட டைரக்டா நீங்க வந்து ஒரு இன்டராக்டிவ் செஷன் வந்து நீங்க கொடுக்கணும் சார். we will also bring your contribution to our management also, சார். சரிங்களா?
ஓகே சார்.
கண்டிப்பா நீங்க வந்து எங்களை நீங்க இது பண்ணணும் சார். சரிங்க சார். ஓகே சார்.
ஷூர், டெஃபினெட்லி சார்.
சார் 2 minutes சார் வந்து இருக்கிறாங்க, உள்ள வர்றாங்க சார், வந்து பேசணும் நாங்க. ஆமா.
சரி, ஓகே சார்.
Hello. Yes, I'm here.
Principal sir. Principal sir.
சொல்லுங்க.
Sir, we are getting a lot of information, sir, from Vasudevan. Isn't it, sir? Okay, sir.
Very good.
Thank you.
சார் இன்வைட் பண்ணீங்களா? இன்வைட்
பண்ணிட்டேன். நீங்க சொன்னது இன்வைட் பண்ணிட்டேன். கண்டிப்பா அவங்க சிவகாசிக்கு வரும்போது நம்ம இதுக்கு வரணும்னு சொல்லிருக்கேன்.
இந்தியா வரும்போது கட்டாயம் விசிட் அவர் காலேஜ்.
கண்டிப்பா சார், கண்டிப்பா வரணும்.
பார்க்கணும்.
கண்டிப்பா சார்.
இப்போ நாங்கள் அட்வான்ஸாகவே உங்களை இன்வைட் பண்ணோம். எங்கள் 25th சில்வர் ஜூப்ளி செலிப்ரேஷனுக்கு.
சரிங்க சார், கண்டிப்பா சார். ரொம்ப ரொம்ப நல்லது சார் வந்துட்டு.
ஓகே ஓகே சார். நீ இந்தியா வரும்போது கால் பண்ணுங்க சார். இந்தியா வரும்போது கால் பண்ணுங்க
சார். கண்டிப்பா சார், கண்டிப்பா சார் கால் பண்ணுறேன்.
ரைட் சார், ரைட் சார்.
I always keep touch with Dr. Vasudevan sir.
Okay.
Sir, shall we go for welcome address, sir? Sorry, vote of thanks, sir.
Okay.
Murugavani, you proceed.
Okay, sir. Thank you, Vasudevan sir, for your valuable sharing. And now it's time to express our gratitude and thankfulness. I request Saran, third year BSc Biochemistry, for delivering vote of thanks.
Good morning to one and all gathering here for this wonderful occasion. I consider it is a great privilege to propose a vote of thanks to all the dignitaries who have witnessed it as a memorable and a successful event. My words are not enough to express the gratitude on behalf of Biochemistry Department. First of all, I would like to say my sincere thanks to our chief guest, Dr. Vasudevan sir, Molecular Genetics and Epigenomic Laboratory, MGEL, College of Agriculture and Related Sciences, Delaware State University, Delaware, United States of America, who blessed us with this thought-provoking conference on international webinar on the science of epigenetics and set a perfect platform to our parents. Thank you, sir, for your valuable speech. Next, I would like to thank our honorable principal, Dr.S.MATHIVANNAN sir, for all words that radiated a source of energy within us. We will ever remain grateful to you, sir. We thank you for being with us today, sir. It's our honor and more than a pleasure to thank our beloved HOD, Dr.S.SUBBURAYALU sir, on behalf of one and all gathered present over here. Thank you, sir. Next, it is a special pleasure to thank our honorable professor who have constantly extended their support to this program. Last but not least, I would like to thank our students who are the integral part of this college. Thank you for giving me this opportunity. Have a nice day.
Thank you so much. Aranya.
Thank you, sir. Thank you.
Yeah, thank you, sir. Okay, sir.
Okay, bye, sir.
Bye.
Good night, sir. Good night.
Good night, sir. Good night.
Sir, shall we wind up, sir? ஃபீட்பேக் வந்துருச்சுலாம்ப்பா. சார் இருக்காங்களா பிரின்சிபல்? இருக்காங்களா சார்? லெஃப்ட் ஆயிடுச்சு. இப்போ ஃபீட்பேக் லிங்க் இருக்கு, போட்டு விட்டுருங்கப்பா.